Well, I'm a research microbiologist. I work in the Division of Healthcare Quality Promotion and a laboratory called the Biofilm laboratory that we started about 12 years ago. I'm actually trained in microbiology and more on the environmental side. I consider myself an environmental microbiologist and a microbial ecologist, really working in the arena of public health and in clinical microbiology. It's been a better understanding of biofilms as microbial communities. We need to understand that it's more than strictly just an organism attached to a surface. The organisms in biofilms interact with one another. And that's important in terms of their ability to attach to a surface, to grow and develop a biofilm structure, and to be dispersed into the environment or into the patient's bloodstream, for example. And how different organisms interact in terms of things like quorum sensing. We've seen the development of new methods for actually examining biofilms directly on surfaces. So we can look, for example, using molecular probes, fluorescent in situ hybridization probes, or various kinds of fluorescent dyes, at methods that will allow us to examine the interaction of cells in a biofilm or examine the effect of a disinfectant or an antimicrobial agent on the biofilm cells. I think we've seen a real development of the use of non-culture-based methods for determining the extent and the diversity of microbial communities, and also, of course, the role of biofilms in things like chronic wounds and understanding that a little bit better. I might use, for an example, a chronic infection would be something that's ongoing, that's sort of recalcitrant to antimicrobial agents. For example, biofilms in osteomyelitis or biofilms on a prosthetic joint or an intravascular catheter. An acute infection might be something that's more easily treated, something that's more easily detected, in the sense that we might have multiple organisms involved in a disease process. And there could be other non-pathogenic organisms that could play a role in the disease process. The whole concept of opportunistic pathogens, organisms like Pseudomonas aeruginosa, which may not be frank pathogens, but can be very important under certain conditions in the disease process. So I think that's an important consideration. One of the things that's really interesting to me is not so much that we find an organism in a biofilm, and even that organism, for example, on a medical device, is related using molecular methods to the bloodstream isolate. But what's really interesting to me is virulence. We do need new approaches. I think the old approaches aren't very effective. One of the things we can do right now, I think, is have a recognition in the healthcare community that biofilms happen, that they're important, that they don't respond the same way that what we call planktonic or non-biofilm cells will respond. So therefore, the first thing might be in the way that we test and evaluate different treatments against organisms. I think it would be helpful to provide new testing protocols that could be standardized, that could be accepted, and could be utilized by healthcare practitioners for treating organisms.